begin. Well, in Dr. Bergella's stead today, I would like to welcome everybody to another in our SMFM Fellow Lecture Series lectures. As usual, we'll have a presentation today by Dr. Ron Wapner, followed by an opportunity for question and answer session. As usual, the question and answers can come from anybody in the audience and can come either as typed in questions or as live chat questions. The way to do the live chat questions is there's a feedback button in the upper right corner, and all you have to do is click that over to the color for Ask a Question, and we'll call on you at the end of the talk, and you can give your question and a very insightful answer, we know, from Dr. Wapner.
Thank you.
We would, if any questions or anything come up, please email me, or you can type in as the sort of the live chat under the question and answer bar, and we can help you out then. Also like to mention the upcoming lectures. Next lecture is on solid organ transplantation, followed in September by infection and inflammation in preterm labor and fetal injury. So I hope you can join in, always the first Wednesday of every month at noontime. I want to thank now Dr. Wapner from Columbia University Medical Center, who's agreed to talk with us today. And he is going to enlighten us on cytogenetics and molecular genetic analysis. Thanks.
Okay. Well, thank you for helping an incapable person get capable. I appreciate it. We're going to, over the next hour, talk about something that someone all of you should know who is illustrated here, had said a number of years ago. This is obviously for those fellows that are really young. You may not even have any idea who this is, but this is Bob Dylan as I knew him, a prophet of the 60s and early 70s, and this is Bob Dylan as some of you may know him. And the reason that I have put that up is because I want to quote from one of his best songs, and it's actually the purpose of the lecture today. And it says, "If your time to you is worth saving, then you better start swimming, or you'll sink like a stone, for the times they are a-changing." And in prenatal diagnosis, and particularly in cytogenetics and genetics, we are in the transition from the older technology, which most of you are familiar with, which is metaphase karyotyping, and using a new term, which is molecular karyotyping. And what I'm going to talk about is how we do this, why it has advantages, and also why it's not time to do this yet, and what the potential problems with this are. So again, we're talking about change. The evolution of what's taken place in the cytogenetics lab has been pretty amazing. When I started doing amniocentesis, and which, by the way, was before ultrasound was invented, so you actually could do it blindly. But more importantly, when I started doing it, this is what a karyotype would look like. And as you can see, the chromosomes are hardly banded, although, as you can see down here, there is some banding. And it was difficult to sometimes even know which chromosome. And we used to argue, is this chromosome 20 or is this chromosome 21? Because remember, they were put in order predominantly by their size. Over the next 20 or 30 years, G-banding, which had just started in this period, really became dramatically improved. And actually, we had not only metaphase banding, we had prometaphase, and a number of ways to differentiate chromosomes. But most importantly, what we did with the bands was be able to look and find smaller areas that either were overrepresented, duplications, or missing deletions, or transposed from one chromosome to another. However, even with this banding at the present time, the best resolution that we have is that we can see pieces that are altered that are about 5 megabases, or 5 million base pairs. And in disease, most diseases, I shouldn't say most, many diseases we now know are caused by much smaller alterations in the genome. We certainly have also moved to fish, in which we can identify presence or absence of very small pieces of the genome. But the problem with fish is you have to know what you're looking for. You can't just fish the whole genome to see whether a patient is missing something. You might say, from what I'm seeing with ultrasound, I would look for something missing on this chromosome. It's been incredibly helpful for a number of things, but the biggest limitation is that you really can't use it to do a whole genome screen. The other thing that we have learned, and again, we used to think human genetic variation was you were missing a chromosome, or you had too many chromosomes, or you were missing an arm of a chromosome or not. We now know that there are a number of different kinds of human genetic variation, and we also know that these different kinds of human genetic variation can or cannot cause diseases. So, for instance, let's take nucleotide substitutions, changing or altering a base pair, and they can be gene mutations that can cause disease, cystic fibrosis, thalassemia, etc. There also can be splice site mutations that are not part of the exons, because we now know that a single gene actually, depending on how it's spliced, can have a number of different functions. And if you have some alterations at those splice sites, you actually can splice it incorrectly and it won't function properly. And we can basically have nucleotide substitutions anywhere along the genome. But, and I think everyone knows that a lot of what we're doing presently with SNPs and everything is we are really looking for normal polymorphism. So between all of this, we do have lots of differences in our base pairs at different locations. They may or may not alter gene function, but they're certainly not disease-causing. So we do know that that's a kind of genetic variation, and indeed, sometimes it causes disease and sometimes it doesn't. Another kind of genetic variation are variable repeat lengths. And we know that if you have a repeat that's too long, it will stop gene function and will cause diseases like Fragile X or Huntington's disease or a number of other diseases that are caused by a high number of repeats. But we also know that repeats are very, very normal, because that's what we use to identify paternity, to arrest criminals, get people out of jail, and that's based on single tandem repeats, or STR. So that can also be a part of the genome that can or cannot cause or not cause disease. The things that we are more familiar with are inversions, in which a piece of the chromosome is cut out, flips over, and placed back in. Everyone's aware of translocations, etc. So what we have learned is that genetic variation is common, and sometimes it's totally normal. It's what differentiates many of us from each other, but in some cases can cause disease. What we're going to talk about today is a specific kind of human genetic variation, and that's what a term that many of you probably have begun to hear over the last year or two, and really was not a term that was used even 4 or 5 years ago very frequently, if at all. And that's variation in the number of copy numbers, copy number variation. Well, what the heck does that mean? A copy number variation is a DNA segment and most people say the segment has to be bigger than 100 base pairs, that's present at an altered copy number in comparison with elsewhere, or a reference portion of the genome. Well, let's just give you an idea, and if you look down here, this would be any chromosome, and we know there's one chromosome from the mom and one from the dad, so any particular piece of DNA on one chromosome would have a similar piece on the other, so the number of copy numbers should normally, as you know, be 2. Here we have an example where in a portion, a bit of the chromosome is missing, so that in some places, rather than having 2 copies, you have 1 copy. And deletion would be a copy number variation, even a very small deletion. Alternatively, if you have 3 copies at a location, that's also an abnormal copy number variation, and that would be a duplication. So when we're talking about copy number variations, you can see that actually trisomy would be a copy number variation, but for a whole chromosome, you would have 3 copies of all the genes. And that, if you extend the definition, whole chromosome aneuploidy is a copy number variation and can be determined as we can others. But most of the things that we're interested in are small segments that have alterations, such as deletions, duplications, and as we'll talk later, these 2, deletions and duplications, would cause disease, but just like everywhere else in the genome, there are areas in which the number of copies vary, so that all of us have certain copy number variations. So let's talk now about the technique or the technology that we use to identify copy number variations. And this is called comparative genomic hybridization, or CGH. And I have a belief, and the older I get, the more I believe it, is that understanding things in nature should be simple. And CGH and how it's performed is incredibly simple, that a 3rd grader should be able to understand it. And again, most major advances really are brilliant in their simplicity. So what one does is we take reference DNA from a supposedly normal person, or apparently normal person, and we cut it up into a large number of small fragments, and then we take a dye of one color or another, and we hook those dyes onto those small fragments. We then, and in this particular example, you can see we hook the small fragments with red dye. And again, it's hooked on by hybridization. We then take DNA from our test person, who we want to know whether or not there is or is not missing material. We break it up into a number of pieces again, and we label it with a dye, but we label it with a different colored dye. We then take these 2 DNAs at the relatively same amount of each, and basically we mix it together. So we now have a mixture of normal DNA that's labeled red. We have a mixture of patient DNA that's labeled green. So the next thing is we have to know whether or not there are equal amounts from the patient and from the standard. So what we need is a target. So we take our mixture of combined DNA and we hybridize this to absolutely normal chromosomes. And actually, as you can see here, the target is and again, I think it's important to point out, this is an absolutely normal metaphase spread from a normal person. And if there is too much reference DNA from our patient on one particular chromosome, that particular area would have more copy numbers and would be red. And if our patient has a duplication, there would be more green, and that would then when you hybridized it, that area would look green. So we take our mixture, we hybridize it to a normal metaphase spread as illustrated here, and this is just an example of an area in which it looks red, and that's because we have more DNA from our normal person than we do from our test person, and this would be what a deletion would look like, because there's an area with less green. Now, that's the way comparative genomic hybridization started, and one actually would look at these metaphase spreads and scan by the eyeball each of the chromosomes. But obviously, that's not much better than what we do looking at metaphase spreads now. So actually, there are computers, and the computer is capable of scanning each of the normal chromosomes, and the readout from for comparative genomic hybridization, normally, if you have 2 copies, it should be equal numbers of red and green. It should be yellow. And if it's yellow, it would have equal copy numbers. Here, we have too much green. That would be a duplication. And here, we have too much red, and that would be a deletion. So the computer reads it. It basically is telling us what the ratio of red to green is. Pretty simple. Here is a sample from a pretty aggressive tumor. So we took DNA from the tumor and we mixed it with normal DNA and we hybridized it to an absolutely normal chromosome 1. And I think the important thing is the targets are normal chromosomes. And you can see there's an area here that's too red. That's a deletion. There's an area here that's too green. That would be a duplication. And it's not uncommon for tumors to have multiple duplications and deletions. But that's how CGH would look. And again, as opposed to looking at different colors on a chromosome, here you can see that it's read by the computer, and there are some areas that's telling us that it's too red, some areas that's telling us that it's too green. That's nice, but it still had problems. Part of the problem was scanning a chromosome and trying to say, is it yellow, red, or green, or is it a little yellow or a little green, is not easy. And more importantly, when you looked at the chromosome, you really had to know what was at that particular site on that normal chromosome. And obviously, the more accurate you were by knowing what your target was, the smaller pieces of missing or overrepresented DNA you could identify. So the next step was to change the target. So this has led to what's called a microarray or array CGH. And basically, rather than hybridizing it to the metaphase spread, you take the normal chromosomes, you break it up into lots of pieces, and you put them on a slide. Now, the important thing is, and the details of this are interesting, but we don't have time to go in it today, when you put these tiny little pieces on slides, you now know where a particular piece of your target DNA came from. So you'd make a slide and you would say, here's a piece from chromosome 1, here's a piece from chromosome 2, but really you would know exactly where on the chromosome that came from. But again, that really gives us fairly good understanding of what our target is and has allowed us to identify smaller and smaller pieces. But the major breakthrough that has occurred has been that rather than taking pieces of target from the lab that are grown in culture, we now actually, because the human genome sequence is known, you actually can take a slide and using technology similar to what you use in plasma TVs, you can say to the computer, I want the sequence from chromosome 1, I just want, and usually these are called oligos or oligomers, and you can build pieces of DNA that are 25 to 50 base pairs. so that that becomes your target. And obviously, the smaller your target, the more accurate you're going to be able to know what's missing. And there'll be 1 million or so sequences built by the computer at each of these sites, and then we hybridize our mixture of red and green to that. And illustrated over here is what an array would look like. And as most of you know, there now are arrays, most of which are used for scientific investigation, that have as many as 2 million oligos on an array, and actually the oligos are picked out for the ones that hybridize best, etc. So really, the resolution that we can query the genome is basically just limited by how many of these oligos we have, and since we know the genome sequence, that's not difficult. So this is interesting, and it certainly has some interest for research, but the question is, what does this do for us as clinicians? Now, this is a readout by a computer of one of those very, very high-density microarrays. But we can mask it so we can tell the computer, I don't want to read every piece. I just want to put I just want you to tell me some pieces from chromosome 1, 2, 3, 4, all the way down to 21, X and Y. So this is from a dense microarray, but it's been masked only to give us a few representative areas from each chromosome. And normally we would do it in the order, so chromosome 1, 2, 3, 4, and the computer knows for each oligo exactly where it came from. So you can see that this would be completely normal. There's no over or under representation, and just ignore this for a few seconds. Here we have a CVS sample, and you read down the array, and you come to where we know there are probes for chromosome 21, and you see that it's overrepresented. And again, there's just a few chromosome 21 probes on it. This would be Down syndrome identified from a CVS using a microarray. But obviously, we can identify much smaller deletions, and here we have a small deletion on chromosome number 8 from band 2-4. So this is how one nowadays reads many of the fine karyotypes that we want to look at. This technology has been incredibly fruitful in the pediatric population. And this just gives you an example of how valuable it is. And this is a number of studies in which they took children who had entirely normal standard karyotypes by what we normally do, but they also had dysmorphic features and or developmental delay. So the parent comes in and says, my kid's not developing, or the dysmorphologist says this kid doesn't quite look right. You say, get a karyotype. The karyotype's completely normal, and the next step has been to do an array. Now, there are arrays depending on how high the resolution of the targets are, but you can see in cases in which the karyotype is normal and in which a kid would have been told he doesn't have a genetic problem, somewhere between about 7 and 14% of those will have deletions or duplications that were identified on array that weren't seen normally. So this gives parents incredible information about why the child had the problem, which is what they're looking for. So arrays do, and again, it's not just of interest from a research standpoint, it really does increase the clinical information that we have. So let's talk not about the pediatric arena, because if you think about it, that's way different. They already have a kid that's dysmorphic or has developmental delay, and they're testing it to find out what's wrong. That's not how we do prenatal diagnosis. We do prenatal diagnosis when we don't know very much, if all, about the kids. So we now are looking at deletions or duplications without having a phenotype. Now, here's an example that should be very, very familiar all of you. We've got an overriding, here's the ventricular septum, we've got an overriding aorta. This is a tetralogy of flow. Now, we obviously could tell the parents, which many people do, look, a TET is fixable. We've got the best surgeons in the world that do operations in our hospital. We'll fix that TET and your kid should be fine. But as you know, we now understand that not all TETs are just a structural abnormality, because on the long arm 22q11.2 there's a deletion, and here's an array that was done on that case that had the tetralogy of flow, and rather than asking to look at each chromosome like I showed you at relatively low resolution, here we're looking at just probes that are coming from the long arm of chromosome 22. And now this kid has a 22q deletion, which, as you know, can lead to DeGeorge syndrome, velocardiofacial syndrome, in which there are a number of other phenotypic consequences. And you say, well, you know, I could do fish. But we're now learning that there are not just one deletion associated with heart defects. We're finding more and more genes, and you have to fish for every one. If you use an array, all you would have to do is take one sample, look over the whole genome. So it gives you precise information of exactly where is missing, and it does a scan or survey, so to speak, of the whole genome. Here is 22q deletion, other phenotypic consequences, and about 20% have moderate to severe learning disabilities. 75% will have a cardiac defect, but 25% won't. They can have palatal, genitourinary, a number of other things, and actually they have a much higher incidence of becoming psychotic and schizophrenic, and many people feel that that number's higher than represented here. So one way the array is helpful is that you can now give the parents a much more detailed analysis of what that what was initially felt to be just a heart defect really means for the kid. But I also want to point out that let's say someday, and this very well is likely to happen, somebody says, we're not going to do standard metaphase chromosomes anymore. We're going to move to microarray. So we could find 22q deletions, and then we do an ultrasound. 25% of the time, they won't have a cardiac defect. 80% they'll have only mild or no developmental delay, but in 20% of the time they will, and again, a whole host of other things. So this really does, if we transition from looking at just cases that have structural anomalies to doing it on all patients, could open up a Pandora's box. And actually, we had a 22q deletion recently in a research study we're doing, and it was really hard to counsel these patients. It's a lot easier when you see something on ultrasound, but as you can see, you always won't. So one of the drawbacks of transitioning to this is the fact that the kid may not have an anomaly on ultrasound, and now you have an abnormality of something in the genome. And the other important issue, there are people with 22q deletions that are as close to normal as can be. And we've discovered these people because they're the parents of kids that have 22q deletions. And there are a number of families in which you're identified because the kid has a problem, and then you look at the parents who appear to be normal, have no heart defects. Most of them will have some palatal changes, etc., but some won't. So we really need to understand the spectrum of these things before we should start testing for them in standard pregnancies. And the other point that needs to be made here, without hopefully belaboring it, these studies have come from kids that were tested because something was wrong with them. So there's a strong ascertainment bias. We need to understand with pregnancies that are just identified with some of these deletions or duplications as part of routine screening, and it's highly likely that the phenotype won't be quite as severe as you see listed here. And I'm not suggesting that this is lethal to this technology. It's just something that we need to keep in mind before we all jump into it. Here's another case we had recently, and just so you know where all these are coming from, we're doing a randomized trial funded by NIH in which we're taking 5,500 pregnancies that are being tested, most of them just for standard indications like AMA or positive screening. And we are doing this to get a population frequency of this. And here we have something abnormal on chromosome 22, but if you look, this is a 22q duplication. And it's highly likely in some of these cases that have lots of deletions that there also will be duplications. They just are hotspots for abnormal crossing over. And just to give you a sense of how these are interpreted, basically now the results are reported as an array on chromosome 22. These are actually the base pair numbers, and a normal case would have ×2, a duplication would have ×3, and a deletion would have ×1. So here we picked up a duplication, and this is way more difficult because here's the mother who's normal. She got through high school and functions as a secretary. One kid has developmental delay and some other phenotype problems. So even if it's the same gene, we just really have to be careful how we use this information in order to give the patients the counseling so they can make the decisions about what they want to do with the pregnancy. Here's another case. So one indication for microarray is obviously testing anomalies so that you can do better counseling. Here's a woman, or actually a blind. Here's a amniocentesis metaphase karyotype. And if you look carefully at the karyotype, you can see that there's a translocation of material from chromosome number 2 onto chromosome number 1. This is a de novo because we tested both parents. Neither of them have it. This would be a de novo reciprocal translocation. And we know that 95% of the time, when you have a de novo reciprocal translocation, the pregnancy is fine. I mean, one piece leaves one chromosome, one piece leaves the other, and you still have normal amount of genetic material. But we also know and have to counsel patients that somewhere around 6% of the time, de novo reciprocal translocation can be missing at one of the break sites a little bit of material. Well, this person was counseled. Actually, Dorothy Warburton, who many of you know is probably the mother of cytogenetics, looked at it. She said, look, as far as I can see, this looks completely balanced. I don't see any missing material. There's a 95% chance this kid should be fine. They made the decision to continue the pregnancy, and at 2 years of age came to see us because the kid had developmental delay and a few other problems. When we now had array technology available, and we looked very carefully, this is chromosome number 2, and you see that there's missing material on chromosome number 2, and that was confirmed by FISH. So another really valuable use of array technology is in de novo translocations, not usually Robertsonian, but reciprocals, to see whether or not material is or is not missing. Obviously, if there's missing material, that's not good. The question is, if it looks like there is entirely normal amount of material, how much does that reduce the risk? And we still have to find that out, because you could have it with a translocation, you could cut in the middle of a gene or a promoter region or something else could be missing. But certainly we do this now on all of our de novo reciprocal translocations, and it has been very, very helpful. It's also obviously valuable if a kid has developmental delay at a de novo reciprocal translocation. Here's something that, for those of you that do a lot of prenatal diagnosis, drives us all crazy. There's 46 completely normal-looking chromosomes, and right here, there's a 47th tiny little chromosome. This is a marker chromosome. That marker could have broken off from any chromosome in the genome, although we know some areas are more likely than others. And right now, when we see that, we basically counsel patients by whether it has satellites or centromeres, etc. And if it's satellites, there's about a 10% chance it will have a phenotypic consequence. If it's not, it'll be about 15%. But, you know, if you knew where that piece came from and you knew what genes were overrepresented, that really would help in your counseling. And this is that case, and we did an array on it, and we see that there's extra material from chromosome number 6, which is a infrequent marker, and then we used whole chromosome painting, in which we took tiny fragments from chromosome 6, labeled them, and then painted the chromosomes. And you can see there's the two normal 6s that are painted, and there is the marker that also has DNA from chromosome 6. So that allows us to know where the markers are coming from in one reaction. Now, you can use FISH, and you can paint the chromosomes, but again, you're guessing where it might come from. And actually, some genetic labs are fantastic. I mean, somebody that's been doing cytogenetics for their life can look at the banding pattern of a marker and say that looks like it's coming from chromosome whatever. But again, this this is way more precise and gives us that level of information. And this is the last example. And this is an amnio, actually this is a CVS that we had done on a patient, and as you can see, there's an X and a Y, and on initial evaluation, it looked like a normal female. However, one of our really good lab techs said, you know what, there looks like there's an extra band in here, and I'm not really sure what that means. And for those of you that have eyes better than I, it looked like that extra band was right here. And the question was, is this an inversion in which a piece came out and was switched over, and that's why the bands are too close to each other, which shouldn't be a problem, or is there actually extra material? And this is very common with even standard metaphase evaluation. At least once or twice a year, the lab will call and say a band looks too big, it looks like something's missing, we don't know, it could be heterochromatin and meaningless, it could be meaningful. So in this particular case, we did an array, and here we're looking at all the probes on chromosome, on the X chromosome, and we actually know where to look because by the metaphase spread, we knew approximately where it was. And you can see that there's duplicated material here. But as I told you, we all have duplicated material, so the next step is first to see if the parents have it. So arrays were done on both parents, and neither did, because in general, if you find one of these duplications or deletions in a normal parent, you would counsel very differently and would not be nearly as concerned. But this is also what the software gives you. And here, this is on the top, the area of the X chromosome in which the material is duplicated, and there's software with all these arrays that tells you what genes are there. So you can see that this is an incredibly gene-rich area. Went back to the literature and found duplications in this area have caused significant problems. So not only can we confirm or deny whether there is an abnormality on a metaphase spread, we can confirm where it is, we can find out if it's gene-rich or gene-poor. So there's a lot of additional information we can give parents. So I think it's fairly obvious that there may be significant advantages in prenatal testing by using this. But before we move this from the lab into standard clinical care, we really have to ask some incredibly important questions. What should a prenatal diagnostic array contain? Should it be one of those incredibly high resolution in which we can find any missing piece, or should we use a targeted array in which we only put oligos or pieces of DNA from areas that we know are clinically significant? How do we handle results that we haven't seen before? There's a lot of missing material, but it's never been reported. So these are really the two critical issues that have to be answered. In the NIH study, we made a decision that we didn't want to use a very high resolution array, but we also wanted the array to be better than you would with standard karyotyping. The reason we didn't want a high resolution array is because we didn't want to be telling people that we find something that's either extra or missing and not be able to know what it is. And our biggest fear is telling people there's something wrong and having people terminating entirely normal pregnancies. So we went with a targeted array, but in addition to picking areas that are known to be associated with microdeletion and duplication syndromes, in order to be equally valuable as metaphase, we also looked at places that didn't have disease-causing or known disease-causing areas in the backbone of the chromosome. So we actually put probes a million base pairs apart, 1 megabase. And again, standard metaphase karyotyping has a resolution of 5 megabases. This is 5 times more dense, but we felt that was actually an appropriate thing. And without letting the data from the study out, we have seen very, very, very few copy number variants that we couldn't interpret for the patients using that. Our array has pericentric regions because marker chromosomes frequently contain a centromere, so this helps us identify markers, and we used 41 telomere regions because I'm sure all of you are aware that subtelomeres, the telomere is obviously the end of the chromosomes, subtelomere deletions are now known to cause mental retardation. So we did 41 telomere regions. We put 70 diseases that have already been described, albeit the phenotype's been described in a pediatric population, which may differ than when you do standard screening of an unselected population. And as I said, we had a backbone of 1 megabase. Also, what diseases do you put on an array? And we decided that in order to be on this array, a significant portion of cases must be caused by deletions or duplications. Remember, this looks for copy number variations, not mutations. So you wouldn't identify the vast majority of cystic fibrosis. Those are usually mutations. So really, we want the diseases to have most of them be a microdeletion or duplication. The disease has to be clinically significant, and we'll talk about that in a minute. We have to really have an understanding of what the full spectrum of the phenotype is, and that I don't think, if we've learned anything over the last year or so, is that we're still learning. And we also, anything that's associated with an ultrasound finding, because that's how we identify these cases, should be on the array. So you can read it as well as I can, but these are the there's 70 of them, but some of the more common microdeletion and duplications that we're using on the prenatal array. Now, as I talked about, there are normal variations in mutations, in repeats, and copy number variants. Actually, variations between normal people and copy numbers is really widespread. 12%, if we compared a bunch of us, 12% of our genome would demonstrate copy number variants, and actually on some chromosomes it's even higher. And the more dense your array is, the more copy number variants you will find. And using a relatively low density array, there's about 12 per person. Now, Most of these copy number variants are smaller than a megabase, and that's why we decided not to look for these. And I think, and those of you who are being contacted, for lack of a better word, by labs trying to get you to start doing more arrays, some of the labs are using 1 million oligos, or some of them are even using 2 million. And if you use those, it's really going to be a problem. So I think a discussion with the lab, and my recommendation is there's really 4 labs or 3 or 4 labs in the country that have done a lot of this prenatal work, and they're using prenatal arrays. I would strongly suggest against using labs that just use high-density arrays, because copy number variants are frequently, they will find them, and then you're going to have to counsel a patient, and there's just not information sufficient to make decisions. In most of us, these copy number variants are normal. They're not associated with pathology. Frequently, they're inherited from our parents. But actually, there's a lot of work now suggesting that they may not be as totally innocuous, it may not be random, that it may have to do with subtle differences in phenotype and, not alter actually gene expression, but modify gene expression. And on this next slide, this is just to illustrate in yellow, you can see these are the common recurrent arrays. The orange ones are ones that are seen infrequently. And for people that are interested, UC San Diego, I believe, has a browser, and also Sanger Foundation has a large project in which every day they're collecting normal copy number variants. And that's going to be really important, because even since we started the study, we now had learned that there are many benign ones. So we can say, we see that all the time, or that's one we haven't seen. So we're getting really good at this. And as I said, we've done about 700 patients in our studies so far, and we have not, this has not proved to be a problem with the array we're using. Here, and I promised I'd come back to this, this is a normal array from amniotic fluid, but you can see that here is some extra material. This is a well-known copy number variant from chromosome 15, and they will show up. So, again, missing material is not always pathologic material. Just to give you a sense of how frequently these occur, if you use, this is again the pediatric series that I showed you that took kids with normal karyotypes, dysmorphic features or developmental delay. And as I said, you pick up about 7 to 10% copy number variants. But those that use the highest resolution arrays, like close to 1 million, 97% had copy number variants, 100%, they found 30 in each case. Those that used targeted arrays, it was relatively low and much more manageable. So basically, when you're making decisions about when and what to use, those are kind of some of the questions you need to understand. I do want to point out, however, that copy number variants are no different than what we struggled with when we went from unbanded to banded karyotypes, because we'd see extra bands and we didn't understand. We struggled with the same thing. Is this clinically important or is it not? And actually, even now in prenatal diagnosis. So it's just unique to looking at the genome. It just can be variable, and sometimes we just don't know what it means, but I think arrays give us the best chance to understand this. In my opinion, arrays will replace standard karyotyping, and I have no conflict of interest with anybody doing arrays. Number one, they have a higher resolution, and more importantly, you do it from DNA. You don't have to grow cells in the laboratory. You don't need to get good metaphase spreads. And how often does a lab say, oh, these weren't good spreads, or, it didn't grow? You don't have to do it, and so it saves a lot of the work. Basically, it's got higher resolution. You get direct mapping of the findings to the specific area in the genome. It's single-step fishing that does the whole genome. But the reason that this will replace things and it is what usually drives things. It's better, but it's also cheaper. I mean, think if you're a genetics lab and you could get rid of your people that scan all your chromosomes, you get rid of all the people that have to look at, grow tissue cultures, and this process can be and is in the process of being completely automated. The turnaround time is just as quick, in some cases as standard metaphase, sometimes quicker. In our study, we have learned that in cultured amniotic fluid in villi, you get enough DNA, but if you take like 10 or 12 cc's of second trimester amniotic fluid, sometimes there's just not enough DNA to do it. So you sometimes still have to culture it, but then analyze it by extracting the DNA. So does have many advantages. And then my last point is a case that to me helped me understand what we need to overcome before we use microarray technology. And in this particular case, I did a CVS, and here is the fetus of the CVS, and it was missing a small piece of the small arm of chromosome 16. This has been described in neonates with seizures. It's been described in neonates with developmental delay. It's also been described with autism and behavioral issues. So that is what's been described when you look at a pediatric cohort. So as I said, when you see a de novo array finding, you look at the parents, and we look at the father. And lo and behold, he has the same thing. He's got a PhD in immunology with the exact same finding as the fetus has. He did report that he has some difficulties as a child with behavioral and learning issues, but, he's done really, really fine, and he's certainly smart. They also, however, had another son who they've been going crazy with because he's 4.5 now. He's got developmental delay. A year ago, he started having seizures. He has pretty much the characteristics that have been described in the pediatric population, and lo and behold, you can guess the punchline, he has the same deletion. So we have in one family 3 individuals going all the way from a kid that does have developmental delay and seizures to a PhD immunologist. So what do you tell this particular family now to do with this information? And that's why I said we really need additional study to know the full phenotype. And actually, we need to begin to understand why it can be expressed relatively differently in some cases. They did make the decision to continue the pregnancy. They said, look, he's got it, he's smart, we can deal with our other child's problems, and they're continuing the pregnancy. So we will know sooner or later. So it is a conundrum sometimes, but in many, many cases, it's given us a lot of information and I think has moved the field forward. So with that, I'll stop. And if somebody tells me what I need to do to answer questions, I'm more than happy to do that.
Great. Thanks a lot for the great talk. I'll open up the floor to any questions that anybody might have. So again, people can type in the questions. People can type in the questions that they want to in the question and answer box, or just highlight the little feedback button, and they can ask them live. Or the talk was so great that nobody has questions.
Oh, come on. Somebody ask me a question. I'm feeling so lonely here. Okay, if you don't ask me, I'm going to ask. How many of you are using arrays in your center right now? And if so, what are your indications and how are you using them?
I think my counselors are in the other room watching, but I don't know if they're they're willing to answer questions. Haha.
Oh, well, okay.
Well, my counselors are there, but they didn't ask a question. I have a question for you. This comes from Cedars. Would you advocate a CGH for all your cardiac anomalies on prenatal sono?
Yes. We actually advocated for most structural anomalies. Now, obviously, it would depend because some structural anomalies are more likely than others. And actually, one of the groups here are doing a research project in which they're doing arrays on all the patients that are seen here and, with heart defects. So we will have additional information. There's also a group doing it with diaphragmatic hernias. And one of the things that we're trying to understand is which anomalies have a high likelihood of being positive and which anomalies don't. So that's still being developed. But with cardiac, should we? I can't answer that. Do we? Yes, we do.
Loyola has chimed in. We're using microarrays for abnormal ultrasound with normal karyotype depending on patient preference. And I think that's what we're doing here a lot at UNC too. So it's only a minority of people that we use it with.
Yeah, and I think that's a really good way to start doing it and get a feel for it. I can tell you the people that we've had difficulty in acceptance hasn't been the docs, it's been the counselors. And what we have heard from the counselors has been, oh my God, we're going to find something that we don't know how to interpret, and it's going to make a very, very difficult counseling session. They've been very, very protective of the patient. So I really recognize that issue, and that's why we do need to be careful of exactly what we find and how we do interpret that.
Our counselors chimed in, but we're not, they were not willing to come on camera. But we've ordered oligos in cases of multiple anomalies and normal karyotypes, depending on patient preference also. I've also used for duplications of unknown origin.
Yeah, and that really is helpful. Okay, well, if there's no other questions,
thanks a lot for
thank you all
for joining us, and we'll see everybody again next month. Again, any questions, please feel free to chime in, and you can do as any time. You can always do it live or as sort of the typed in chat. And if you have questions about any of those, feel free to email me. My email's on the website. Thanks very much.
You're very welcome, and thanks everybody for